We are here with Homayoun Bagheri. He is professor for evolutionary biology. He has some fantastic research. Welcome, Homayoun, to the show. What is evolutionary biology?
Well, that's a good question to start with. I think that evolutionary biology tries to answer some of the questions that have been at the core of what humans have been asking about themselves and the world around them since we've had people with the ability to ask scholarly questions, so to speak. So, what are the basis of living organisms? Why are we here as human beings? What is our relationship to these other organisms? And these kinds of questions have occupied philosophers and scientists from before the Greeks, essentially, before the ancient Greeks. And I think one of the watersheds or the revolutions that happened in our way of understanding nature, or at least living organisms around us, was towards the end of the 19th century when Darwin came with the Origin of Species, and that helped us have a more objective way of looking at nature and how organisms are designed.
How have we looked at nature before Darwin, and how have we looked at nature after Darwin?
Excellent. So that's interesting. If one really wants to look at it from a historical perspective, I think that initially we were just happy with classifying what's out there. If you look at the biology that was done before Darwin, essentially you had colonial expeditions that would go out there and classify and catalog the organisms out there, and there was no big ambition to understand the logic or the scientific, the causal processes that led to this diversity of life, other than, of course, you would have people who would assign a deity to the design and reproduction of all these different organisms, essentially. And with Darwin, what we had was one of the first times where we tried to successfully apply a scientific method based on objective criteria on how you could understand the diversity on the planet, and more importantly, from my perspective at least, is why organisms are designed the way they are. How do they come about to manifest the particular designs that they manifest to us?
Now, you look at very simple organisms, and you look at a transition, a very important transition from single cells to multicells. Tell us a little bit more about that.
Sure. Well, to be honest, I would love to look at more complex organisms, but to understand some of these, what people sometimes refer to as major transitions in evolution, essentially events in evolution where the organizational structure of an organism changed drastically, and often it led to a higher level of complexity. One example being going from single cell to multicellular organisms. And we would really like to understand what are the processes that underlie these transitions in evolution, and to start to understand these in more complex organisms would be a very difficult challenge, essentially, and it still is a difficult challenge to do it with simple organisms. So the idea is that if we go on the basis of the assumption that, or essentially the deduction that we've had empirically, that all organisms are related to each other. So we want to understand the series of processes that have led to the early simpler organisms, to the complex organisms we have today, such as you and I. We have to understand what governs these transitions. And one of the most important is this transition to multicellularity, because once you go from a single cell to a multicellular cell, you suddenly are exposed to a whole set of new advantages that you can use, essentially.
What are these advantages? If you have, let's say you have an organism with 3 or 4 or 100 cells, which is still very small. What are the advantages as opposed to being a single cell?
Yes, so that's excellent. So one has to be very careful and not to mix issues. So often people have mixed, for example, multicellularity and division of labor as one transition, while one should probably separate these issues. So first you have on what people refer to as undifferentiated multicellularity. So you have a bunch of cells that stick together, but they are all the same. right? Somewhat like in a social context, like you refer to a mob of individuals all having the same opinion, for example. The advantages of this simpler form of multicellularity would be, for example, you would have a larger size, so you can either avoid predator or be a better predator. You can have higher efficiency of feeding, for example. While if you start having a more complex form of multicellularity, such as division of labor, then as you also have in society, as we see around us, for example, you can start having cells specialize on different activities and getting a higher efficiency of metabolizing the food around you or producing motility or other structures that you otherwise would not have.
So just like us, we
Yes.
have division of labor here.
Yes.
We have
Yes.
skin cells, and we have
Yes.
liver cells, and
Yes.
we have eye cells, and
Yes.
and brain cells.
Yes.
So that's division of labor, and you see that in the
Yes.
very smallest multicellular organisms already.
Yes.
Even an organism that has like 2 cells, do you see that there is a division of labor?
Almost. So what's fascinating, if you look at the history, I mean, in a sense, evolutionary biology is also a historical activity. We're trying to understand the history of the planet, essentially. We are historians in some sense in natural sciences. If you look at the age of the planet, it's about 4.5 billion years old, and probably the first manifestations of life we have in the fossil record are about 3.5 billion years old. So this is a long time ago, right? And in these very first microfossil manifestations, one already sees evidence for multicellular organisms that were already present on the planet. And maybe this division of labor we're talking about, at least the evidence we have for it in the microfossil record, maybe about 1 billion years later, maybe slightly less, you started having division of labor, that you could actually see it in the morphology, that you would have essentially filaments of bacterial cells that are stuck together, that form essentially a tube of multicellular entities. And you would have different kinds of cells, ones that would do photosynthesis, which is essentially taking the energy from the sun and converting that energy or using that energy to produce sugars, carbohydrates. And the other cells would fix nitrogen, essentially take free nitrogen and use that to make some of the components that you later use for making proteins. So in a sense, these are two of the most important biochemical reactions that we have on the planet. Without these biochemical reactions, life on Earth as we know it would not have occurred. Because with photosynthesis, suddenly you open up a new channel from where you can harvest the energy that is hitting the surface of the planet through the sun and use that energy to construct biological material, biomass essentially. And you also needed the nitrogen fixation to make the proteins with which you make cells and the machinery within these cells.
Now, your research, you use what kind of organisms to play around with this and then measure and see what's happening in multicells?
Yes. So given that the evidence from the microfossil record shows that some of the earlier organisms were filamentous bacteria, we are interested in understanding these filamentous bacteria, understanding what governs their multicellular structure, what can lead to the evolution of multicellularity, and what can lead to the loss of multicellularity, for example. And I think this is a classical case where one can also illustrate some of the difficulties and challenges of trying to understand evolutionary history. After all, we want to understand what happened 3 billion years ago or 2 billion years ago. And since we don't have a time machine and we can't travel back and see what happens, we have to develop some scientific tools to do that. And essentially there's 4 tools that one uses to do that. One is that we have a fossil record. In my group we don't work with fossils, but of course we use the evidence that other paleontologists would have discovered. 2, you want to look at the genetic material in any kind of organism you study. In our case, we're studying these very simple bacteria, essentially. So you study their genetic material in order to understand their evolutionary history. And by that, what I mean is, maybe I can make an analogy. If you are a linguist and they present you 4 books, for example, you have Don Quixote and you have something from Gabriel Garcia Marquez, for example, and then on the other hand, you have something from Shakespeare, and then maybe, I don't know, Milton or whatever. Okay, and you want to understand what is the relationship between these four books. You look at the language inside and you find that the two books in English have many similarities, while the two books in Spanish have several similarities. And from that you construct what you refer to as a phylogenetic tree of the relatedness of these books, or the content, the cultural content of these books based on the language. So one does something similar by looking at the genetic content of organisms. So you look at which genes are coded in these organisms, what are the similarities and differences in gene content, and you build a phylogenetic tree. And this is important in order to understand the history of these organisms, and we get some empirical data. The third thing you can do is you can start doing experiments in the lab. So if these organisms have maintained some of the properties that they also had in the past, then if we apply some of the same forces of natural selection, for example, or environmental forces that change the environment around these organisms, you could see the effects that these forces would have on changes in these organisms. So people do what is referred to as, for example, experimental evolution, where in the laboratory you would try to select for properties that you might have also seen in the natural environment, and see if selection is successful in evolving new characters. One thing we do, for example, is we're selecting for the loss of multicellularity, because once you select for organisms that have lost multicellularity, you can look at their gene content and see which genes were damaged, for example, which genes were lost, in order to understand what is the machinery involved in multicellularity. And fourth, and this is something that we focus on a lot in my group, is to do theoretical models, essentially. And this is not very different from what theoretical physicists, for example, would do, which is you would construct mathematical models based on some of the assumptions that you have about the phenomenon you're studying. In our case, of course, the assumptions will be based on some of the empirical studies that biologists have done, or we ourselves have done. You put all these assumptions, for example, by assumptions I mean, for example, you say, if A then B, if B then C, right? But this is much more mathematical when we do it. And you put, yes.
Yeah, sure. One question. So you said on point 3.
Yes.
You know, multicellularity can be lost again.
Yes.
So this has happened several times in the evolution of the planet, of life.
Yes. Yeah.
Okay, so they go back and forth. They go from single cells, they build filaments, and then eventually they lose it again, and they build it. Why do they lose it again, and why do they build it again?
It's an excellent question. I mean, multicellularity has been one of those questions where people usually have had many interesting ideas about how multicellularity evolved. But usually, we as scientists, we've started with a lot of pre-assumptions, right? That it has to make sense that you start with very simple organisms that are single cell, then you become multicellular and further complex. And with this assumption in mind, we've sometimes ran along with that. But interestingly, when, for example, we try to reconstruct the phylogenetic history of the cyanobacteria, for example. One of my PhD students did her work on this. It turns out that what you see is that early in the origin of the cyanobacteria, you had very simple single-celled cyanobacteria, but very early on, the multicellular filamentous cyanobacteria evolved, or multicellular filamentous ancestors. And this lineage of multicellular bacteria was the successful lineage that essentially colonized the planet, according to our phylogenetic analysis at least. And then once the planet was colonized by these organisms, in certain locations they lost their multicellularity again and became single-celled when they were specializing in an environment or not changing their environment, presumably. So once you look at the empirical data, the surprises that you always get in science is that some of your assumptions are going to be destroyed. You look at and you get counterintuitive things, like, as I was giving the example, that multicellularity evolved very early on and then it was lost several times. And this is a bacterial example. And there are other examples in algae, for example, that where one sees that, again, in multicellularity was lost again. And there are many other kinds of transitions where there's been a reversal in complexity in biology. If you look in the social insects, for example, the insects which form complex society, hives and things like that, you see that eusociality, which is a name for some of the more complex social structures, is lost several times in some insect lineages.
So knowledge can get lost in a sense. I mean, you could tags this as knowledge, and it could get lost, because we go under the assumption that if something in society, if something has been developed, if a technology is out there, it's out there, it can't be lost. You know, if a book is written, it survives. But maybe a wrong notion. So knowledge can be destroyed.
That's a good metaphor for it. Technically speaking, we could say that information is destroyed, right? The information that is coded in the genes is lost or destroyed, the same way as knowledge can be destroyed or lost in societies. Yes. So that's actually a very nice metaphor for this. Yes, absolutely. So,
and going back to this, you know, multicellularity, single cellularity. If we go back in our history, back until we were, you know, single cells, have we gone back and forth also a couple of times, single cell, multicell, until finally we became super multicell thing?
Most probably. So one of the reasons I'm interested in multicellularity is that because I want to understand how complex organisms such as us, mammals or human beings, evolve. And to do this, you try to look for generalities, the rules that lead to the evolution and loss of multicellularity. Because when you look at the different kingdoms in life, you know, plants, animals, bacteria, what you find is that multicellularity is being gained and lost in several lineages of this tree, at least 20 times for very different organisms. So this is happening all the time, or happened all the time during evolution. And presumably it also happened in our history. The problem is that figuring out the early ancestor of the metazoans, we are within the lineage of what are referred to as the metazoans, has turned out to be quite difficult, because this is a very deep root in the eukaryotic tree, which we are eukaryotes, and that has not been resolved yet. But when you look at the organisms that are at the bottom of this tree, you know, things like sponges, for example, you know, these are very simple multicellular organisms, and we know that there are lineages where they've lost it and lineages that they're gaining multicellularity. But you know, as human beings, you know, or as mammals, we are very far away from that very early origin of multicellularity. I think what made us much more special is the kind of division of labor that subsequently evolved in our developmental plan, essentially, where at least, and that's also actually not specific to just mammals, right? So there's many vertebrates with high level of division of labor. You also have invertebrates that have a high division of labor. And this seems to be a sort of signature of what you refer to as complex eukaryotes that have evolved a very high level of division of labor.
And what makes that? I mean, the question is, I mean, we are extremely complex. But you have your model organisms, cyanobacteria is quite a simple organism. At what decides that something stops evolving complexity, and what makes something develop complexity? Have you figured that out?
Well, that's a fantastic query, so to speak, because this was one of the puzzles for us. We know that bacteria have been around, they've been around at least 1 billion years before the eukaryotes, right? At least, if not more. And they owned the planet for a very long time, and they evolved multicellularity at least 1 billion years before the eukaryotes. And what is it that stopped them from becoming more complex? Why are you and I not bacteria? Why are we not multicellular bacteria? And there's some explanations, possible explanations for that. One is that to evolve something as complex as you and I, you would require to have genetic machinery that can have the programming, so to speak, if I may use a metaphor, that can have the programming capabilities to manifest all the different kinds of cells we have, and also the capabilities to construct some very complex biological organisms from a single-celled organism, right? Development is a magical process where you start with a single cell and you create something like adults like you and I. Yes, I do not want to keep saying that we are complex, but yes. And you need a very complex genetic machinery for that. And when you look at bacteria, first of all, their genomes, in other words, the number of genes they have in the gene content is much smaller. They have much less genes. Let's say on average, you might, in a mammal, you might have about 30,000 genes, while in bacteria maybe 1, 2, or 3,000, sometimes up to 6 or 9. Secondly, it's not only the number of genes, but how you regulate these genes, and by that I mean how you say, okay, these two
turn
genes
them on and off.
Yes, exactly. How you turn them on and off, and the machinery for that is much more complex in eukaryotes. Thirdly, the cells have become much more complex in eukaryotes, and this is actually a good example, I think, to illustrate the sort of a realization that when we look for increases in complexity in life, what we see is often that you have two independently evolved entities, whether they're organisms or some biochemical pathways, and by combining these together, you form a more complex new entity that can do something that neither of them could do before. An example of that is the eukaryotic cell, which we now understand that they evolved from what is referred to as endosymbiotic event, which means that you have a host cell, some kind of bacteria or archaea, which engulfed another bacterium essentially, and together they formed a new kind of cell that had more capabilities. In other words, they also did a division of labor, so to speak. You also see it at the biochemical level. The oxygenic photosynthesis we see today is much more efficient than the photosynthesis the very older bacteria did, and that's also a combination of a couple of enzymes that are able to do this.
Now, looking forward in, let's say, in 10 or 20 years, what is your big dream? What is your big thing you want to have accomplished in your research?
As a scientist, I try to be humble and try to use, the tools that are available to us to try to understand something that is real and something that will not be sort of discounted 20 years down the line to say Bagheri was wrong when they discovered this. So within this framework, I would very much like to understand what are some of the ecological and evolutionary forces that govern a transition, such as the transition from single cell to multicellular organisms. And the hope as a scientist is always that these model systems that you're working on, whatever it is, you put a lot of effort in understanding the details of that system, but your ambition is always that what you're finding there, you can extract generalities from that process so that you can apply those generalities to other model systems that will be undergoing a similar transition, in my case, for example. So understanding what will lead to the increase in complexity in an organism, multicellularity being one example, would classify as one of these dreams, as you're asking me about.
Great. Yes. Okay, well, thanks so much for your answers, and good luck with your research.
It's been a pleasure, Rolf.